Hello friends, let us now learn some important points about the dermatology. In the dermatology, in this chapter, we are going to learn about the basics of dermatology. In this, first, skin is the largest organ of the body. The weight that skin has is 4 kgs and the skin constitutes 17% of total body body weight. Skin constitutes 17% of total body weight. The area of skin is 1.7 square meters. Now, the skin has many units. It has epidermis followed by dermis. In between epidermis and dermis, we have dermoepidermal junction, and the last layer is subcutaneous tissue. Now, first we will see some important things about reactive units. So in the reactive units, first I will draw a picture so that we will understand what a reactive unit is. So this is the skin, and the upper part is epidermis. Then epidermis and dermis is separated by dermoepidermal junction in between, and then the lower one is subcutaneous tissue. So, and in the dermis we have 2 layers of blood vessels are present. So one is superficial vascular plexus and we have deep vascular plexus. 2 layers of blood vessels. One superficial vascular plexus, second deep vascular plexus. So if you see, that is in the dermis, the area of dermis which is above the superficial vascular plexus is papillary dermis, and the area of dermis below the superficial vascular plexus is reticular dermis. So this is superficial vascular plexus and this is deep vascular plexus, and we have epidermis and you have dermis, and below it, that is subcutaneous tissue. The area between the epidermis and dermis is dermoepidermal junction. Now what is the reactive units? We have 3 types of reactive units. One, we have superficial reactive unit which starts from epidermis, dermoepidermal junction and papillary dermis. Together we call it as superficial reactive unit, whereas reticular dermis is only called as deep reactive unit, and the subcutaneous tissue is called as subcutaneous reactive unit. Now, if you see, this is the subcutaneous tissue with subcutaneous reactive unit. In this we also have the hair tissue is present. So that is the hair tissue which arises from the subcutaneous tissue, and there is also a sweat gland which is associated with the hair, which opens in the hair, is also seen in the skin. Okay. Now, in the subcutaneous reactive unit consists of some components. So the components of subcutaneous reactive unit include hair, subcutaneous tissue, nail, glands like sebaceous glands and sweat glands. Then this is about the reactive units. Then we will learn first about the first layer of the skin. The first layer of the skin being the epidermis. So first, epidermis. In the epidermis we will learn about the cells present in the epidermis, which include keratinocytes. There are 4 types of cells: keratinocytes, which are most common, melanocytes, Merkel cells and Langerhans cells. So first, in the keratinocytes, these are actually most common, and these keratinocytes constitute around 85% of cells in the epidermis. Then we will learn about keratinocytes. Keratinocyte is derived from, this keratinocyte is the cell which is present in the skin, and it is in the keratinocyte, as the name says, it is presence of keratin filaments, or there is presence of tonofilaments or intermediate filaments are present. So that is the cell, and here in the cell you will see presence of keratin filaments. So this presence of keratin filaments or tonofilaments or intermediate filaments, all these we together called as keratinocytes. Now keratin can be either soft keratin or hard keratin. So if you see the keratin, it can be either soft keratin or hard keratin. This soft keratin has less disulfide bonds are seen in soft keratin. That is, for example, this soft keratin is present in skin, whereas in hard keratin you will see presence of more disulfide bond, and it is present in nails. So these are disulfide bonds which are present in nails. Then I have said that this keratinocyte contains keratin filaments. So we will learn some important points about keratin filaments. This keratin filaments is a heterodimer. It is a heterodimer which contains keratin subunits. So it has K1 to K18 subunits. So for example, if you see either K1 or K18, so these combinations are present and they form keratin filaments. So they will be from first K5 to K5 or K14, both these will combine together to form then this a keratin filament, and this is present in stratum basale. Then K1, K2, K3, K4, K5, K6, K8. This is how I remember. Then after K8 it starts from K10, 11, 12, K12, then K13, K14, then K16, 6 for 16 and 8 for 18. So this is how I remember. So first K1, K10 it is stratum spinosum. Then K2, K11 it is stratum granulosum. Then we have K3, K12 it is stratum corneum. Then K4, K13 buccal mucosa. K5, K14 stratum basale. Then K6, K16 it is present in nails. K8, K18 in liver. So if you see, this is the important table which we should remember. Now if you see, so this is how I remember it. Always K6 and K16 will be nails and K8 and K18 will be liver, and then it is from reverse: spinosum, granulosum, corneum, basale. Okay, so I don't know, you can make any type of mnemonics that you want and you should remember this. Now if you see, this keratinocyte is derived from ectoderm. Now if there is a defect in K8 by K18, then this will result in liver cirrhosis. If there is defect in the stratum basale K5 and K14, it might result in epidermolysis bullosa simplex. So the next important cell in epidermis is melanocytes. So these melanocytes are if you see, the keratinocytes are derived from the ectoderm. K for K, that is ectoderm for keratinocyte. Whereas if you see the next cell, which is melanocyte, this melanocyte is actually derived from neural crest cells. So let me draw a melanocyte. A melanocyte has many the dendritic like processes. Then this is melanocyte, and it is surrounded by many keratinocytic cells. So these cells are keratinocytes, right? And the nucleus of keratinocytes. And now if you see, this melanocyte is derived from the neural crest cell M followed by N. So it is derived from neural crest cells. The melanocyte is the most common basal layer or stratum basale. It is present most commonly in stratum basale or basal layer. Every 10th cell is a melanocyte in stratum basale. Epidermal melanin unit constitutes 1:36. That means there are 1 melanocyte supplies with 36 keratinocytes. That is, 1 melanocyte is surrounded by 36 keratinocytes. And the main function, so this epidermal melanin unit is 1:36. Right, that is, 1 melanocyte is surrounded by 36 keratinocytes. Then if you see the next important thing here, the melanocytes help in synthesis of melanosomes. Once melanosomes are synthesized, those will travel through the dendritic processes and reach the keratinocytes. So the synthesis of melanosomes occur in melanocytes, and these are supplied to the keratinocytes, and this will impart the color to the skin. That is the reason why we have different shades of skin color are present in different people. So this different shades of skin color in the people mainly depends upon the melanosomes mainly. So if you see, the different shades of the skin color depends upon the two things: one, differences in size, number and distribution of melanosomes, and it also depends upon the differentiation in differences in proportion of eumelanin and pheomelanin. So based on the different shades of the skin color, a classification was put forward by a scientist called as Fitzpatrick, and this is called as Fitzpatrick's skin classification. And this classification, Fitzpatrick's skin classification, is started from 1 to 6. 1 is never tans, always burns, whereas 6 is always tans, never burns. And this Fitzpatrick's classification, so Fitzpatrick's, for this classification, he is called as father of modern dermatology. Then the next important thing is the defects. So whenever there is defect in the melanocytes completely, so whenever there is defect in, that is absent melanocytes, it is, if the melanocytes are absent, then the disease is vitiligo. If there are increased melanocytes, if they are increased locally, then we, that is at a particular site in our body, then we call it as lentigenes, where you see hyperpigmented macules. Then if there is defect in the melanogenesis, that is, first, whenever there is defect in transfer of melanosomes, that will cause nevus anemicus. Where there is defect in transfer of melanosomes, where it causes nevus anemicus or nevus depigmentus, right? That is defect in transfer of melanosomes. Then if there is defect in melanogenesis, if there is complete defect in melanogenesis, then this will cause albinism. Okay, that is wrong, it is only albinism. If there is defect in melanogenesis, it causes albinism, that is, there is no skin color imparted. Okay, if there is increased melanogenesis, then this results in melasma or freckles are seen. If there is increased melanogenesis will result in melasma or freckles. So these are the important things. Then the third important cell which you have to learn is about Langerhans cells. This Langerhans cells belong to a family which is called as dendritic cell family. So these are antigen presenting cells, which are, if you see, this is this Langerhans cell contains large and many dendrites are seen, and in the center you will see a nucleus, right? So this Langerhans cell is mainly derived from the bone marrow. It is derived from bone marrow and it secretes interleukins also. It is derived from bone marrow, it secretes interleukins, and these Langerhans cells are constant in different parts. So one important thing is these granules which you see in Langerhans cells are very important. These are seen on electron microscopy. These are called as Birbeck granules, or we can also call it as tennis racket granules or rod shaped granules. So these granules are used in receptor mediated endocytosis. These are mainly used in receptor mediated endocytosis. Now if you see the immunohistochemical markers, the immunohistochemical markers for Langerhans cell are very important. Langerhans cell are actually positive for CD1A, CD207 and CD100. CD207 codes for Langerin, and they are negative for CD14, 68 and CD163. Now if there is any defect in the Langerhans cells, then that is, if there is increased Langerhans cells, it leads to histiocytosis X. If there is decreased Langerhans cells, it causes psoriasis, sarcoidosis, and you will also see contact dermatitis is seen. Okay, that is given by the mnemonic PSC. Next is increased Langerhans cells. It causes a histiocytosis, which is of three types. One, eosinophilic granuloma. Second, Hans color Christian disease. And third, Litterer CV disease. In first, eosinophilic granuloma, if you see, eosinophilic granuloma is seen in less than 8 years of old child, and Hans Killer disease less than 5 years, and Litterer CV disease less than 2 years. Now in eosinophilic granuloma, you will see presence of lytic lesions in the bone, temporoperital, and in the bone, that too most commonly lytic lesions are seen in the temporoperital area of the skull. Whereas in Hans Kuller Christian disease, which is seen in children less than 5 years of age, you will see that there are triad, that is multiple bone defects, diabetes insipidus, and exophthalmos triad is seen in Hans Kuller Christian disease. Then if you see, Litterer CV disease is seen in less than 2 years of old child, and this shows generalized fulminant condition where you see bone, skin, and hepatosplenomegaly can occur, and even you can see presence of seborrheic dermatitis may be seen. Then the fourth and last cell which you see here is the last layer of cell is Merkel cell, which we will learn later. Thank you for watching.